Hey, this is Rochelle, your Microbiology IT tutor, coming back with another viral genomic video. This is part 2. If you want to follow along, you can download the fill in the blank notes. The link is in the description box. Feel free to subscribe if you want more videos like this. Like the videos, share it with your classmates, and hey, feel free to leave a comment. Let me know that you're learning something. All right, let's microbiology it. DNA archaeoviruses are a unique group of viruses that infect archaea microorganisms that thrive in extreme environments such as hot springs, acidic lakes, and deep sea hydrothermal vents. These viruses primarily have double stranded DNA genomes, although some single stranded DNA archaeoviruses also exist. Unlike bacteriophages and eukaryotic viruses, DNA archaeoviruses exhibit a wide range of unusual morphologies, include bottle-shaped, spindle-shaped, and even two-tailed structures, which are rarely seen in other viral groups. One well-known example is Pseudophilobus terete icosahedral virus, STIV, which infects hyperthermophilic archaea and has structural similarities to eukaryotic viruses, suggesting a potential evolutionary link. DNA archaeoviruses replicate using various mechanisms, often relying on host enzymes due to their compact genomes. Some integrate into the archaeogenome as prophages, entering lysogenic cycles, while others use lytic cycles to release new virions. Because archaea live in extreme conditions, these viruses have evolved unique stability and replication strategies, making them valuable for studying virus evolution and adaptation. DNA archaeoviruses have double-stranded circular DNA genome. Uri archaeotaviruses are methanogenic, and halophilic. So we're going to fill in the blank here. They have a head and tail type similar to phage T4. Now we have host number 1, a sulfur chemolithotroph. Pseudophilobus. And the virion is SSV. It has a circular DNA genome and it is found in acidic hot springs. Another virion is SIFV, and it has a linear DNA genome. Our second host is hyperthermophile acidimus. The virion is ATV. It has a circular genome, production of long, thin tail at each end of the virion once released from the host cell. The virion becomes thinner, the volume is reduced, and has a lysogenic cycle. Our third host is hyperthermophile pivococcus. The virus is PAV1. It has a small circular DNA genome. It's released without the host cell lysis, usually in a budding mechanism, and it is extremely heat stable. A possible origin of single-stranded plus sense RNA viruses can be found in hot springs, and they encode for RNA replicase. Now, DNA animal viruses, pox viruses. We have Edward Jenner. He created the pox vaccine. He exposed people to less virulent cowpox to protect them from smallpox. So that was Edward Jenner. Other viruses under the DNA animal viruses are cowpox and Vaccinia viruses. Vaccinia viruses are not pathogenic. They are used as a smallpox vaccine. So smallpox. They have a linear double-stranded DNA. Replication occurs in the cytoplasm of the host, and they are genetically engineered to have certain proteins for the use of vaccines. Adenoviruses, they are small and naked icosahedral viruses. They have linear double-stranded DNA genome, and they cause mild respiratory infections in humans. So we're going to do like a little illustration here where we have the adenoviral genome. Oops. DNA here. That's what that is. And then we are at the 5' end here. And then this section here, we have the adenoviral terminal protein. And this is essential for DNA replication. Transcription in the host is in the host nucleus by the help of the host RNA. RNA polymerase. There are two proteins that are transcribed. It's the terminal protein and the viral DNA polymerase. The terminal protein binds to cytosine, which is the primer for DNA polymerase. The initial replication products are the complete double-stranded viral DNA and single-stranded minus-sense DNA. DNA tumor viruses. Polyomavirus SV40 is a naked icosahedral virus. It has a circular genome, double-stranded DNA, and causes tumors in small animals like hamsters and rats. The host DNA polymerase is used during viral replication. Let's move this up. Transcription happens in the host nucleus. Virion assembly is in the nucleus. And the host cell lyses and releases the virions. Now, viral infection, the formation of new virions, and then the host cell lyses to release virions happens in these types of host, and it's called permissive. Here, there's a viral infection, and then the viral DNA is inserted into the host DNA, and the host cell is altered. And these types of hosts are called non-permissive hosts. So there is, in this process, there is a loss of growth inhibition, and the cell becomes malignant. And this is called transformation. So the host cell is transformed. Transformation. And then the tumor cell-induced proteins promote uncontrolled cell development by binding to the host cell proteins or inactivate the host cell protein. So it's tumor cell-inducing proteins. A herpes virus will be our next topic. They have double-stranded DNA and they are enveloped. The human diseases associated with herpes virus are fever blisters, venereal herpes, chickenpox, shingles, and infections, mononucleosis. Cancer-causing virus diseases: Epstein-Barr virus, Burkitt's lymphoma. Our next virus is cytomegalovirus. Cytomegalovirus. There's no apparent symptoms, and they have a long-term health consequences. The disease associated with CMV is pneumonia, retinitis, and certain gastrointestinal disorders. With CMV viruses, they can be latent. In a long period of time, they can exist in the host. They can become active under stress or active when the immune system is compromised. Viral infection. How does this work? 1, there is an attachment by fusing to the host cytoplasmic membrane. 2, there is the release of the nucleocapsids and they are transported to the nucleus. 3, there is the uncoating of the DNA, and 3 classes of messenger RNA are produced. They are 1, the immediate early, 2, delayed early, and 3, late. And I will go through each one. So we have immediate early. The messenger RNA encodes for regulatory proteins that stimulate delay early proteins. Delayed early is proteins that are synthesized. Now we have the viral specific DNA polymerase and the DNA binding protein. Lastly, we have late. It is the viral structural proteins. 4, rolling circle replication produces concatenames. Thank you for participating. Don't forget to subscribe, like, share, and leave a comment. I would love to hear from you. This is Rochelle, your microbiology tutor.